Good evening, this is Mr. Wintermelon. So I have stopped my editing in esophagus, and in this video, you will learn about digestion of food in stomach and beyond. So after our esophagus, the bolus will arrive in the stomach. A stomach is a distensible bag, that means it can be stretched. It is very muscular. And the stomach have numerous pits, which leads to gastric glands that secrete gastric juice. So our muscle will contract and relax, and this will churn up the food, something like you can imagine witch is mixing her potion and so on. So the food is mixed with the gastric juice for chemical digestion to take place, as well as the churning of food will mechanically break down the food even further so that the enzyme have a greater surface area to add on the food. And just for your info, there's a thick layer of mucus that lines the internal wall of the stomach. And you might be wondering, mucus? Isn't those things only found in our nose? No. It is also found in our windpipe as well as inside our stomach, and there's a reason for this. So this is more of just for your personal knowledge, more of extra information. Inside our stomach, there are cells that secrete hydrochloric acid as well as the enzyme known as pepsin. These two actually you must know. Stomach or gastric glands secrete hydrochloric acid as well as pepsin. Then there's also mucus that is being secreted by the stomach, and the main purpose is to protect the wall of the stomach from the hydrochloric acid, because it is a strong acid after all, and the pH of a stomach is 2. So you need this mucus to help to neutralize the acid so that they would not damage the wall of the stomach. So as mentioned, gastric juice contain hydrochloric acid as well as pepsin, and pepsin is an enzyme that digests protein. That's why it's a protease. So the purpose of hydrochloric acid, besides making the environment of the stomach to be acidic, which is pH 2, this will allow the activation of pepsin as well as to stop the action of salivary amylase. But this one, I wouldn't say that it's a function, it's more of a consequences. So the hydrochloric acid will help to denature the protein that we have consumed. And as you have learned in the previous chapter, protein can be denatured by extreme pH. So some of the protein that we have consumed will be denatured by the acidic pH of the stomach, and this will unfold the protein, allowing the enzyme to have a larger surface to act on the polypeptides. The hydrochloric acid also helps to kill bacteria and germs. As for pepsin, it breaks down protein into shorter chains of polypeptides, and the acidic pH definitely helps to speed up this process. As mentioned, some of the protein would be unwind, which make it easier for the pepsin to add on. So after about 4 hours or so, the food will become more liquefied, and it is now known as chime. So chime will be released into the small intestine through the pyloric sphincter as it opens and closes. So now you can see that stomach actually is quite useful for storing food because it stores food there for 3 to 4 hours. This is important as it gives time for the enzyme to chemically break down the protein into polypeptides. Now into the small intestine. So it's important to understand the structure of our small intestine. Our small intestine is actually very long. It is 5 to 6 meters long, consists of 3 regions. The name of the region you need to be familiar with. The first part is called duodenum, followed by jejunum, as well as the last part, which is the longest part, known as the ileum. So the inner surface of the small intestine is extensively folded, forming villi. The wall of the villi is thin and consists of only one layer of epithelial cell. That means the wall of the villi is only one cell thick. The duodenum, or the start of the small intestine, will receive bile from the gallbladder as well as pancreatic juice from the pancreas. Those are the accessory organs in our alimentary canal, which means food doesn't really pass through them, but they definitely helps in the process of digestion. So let us take a look at the accessory organ. So we have our pancreas that secrete digestive enzyme at the pH of 8.6, so it is slightly alkaline. Liver also produce bile at a pH of 8. Then we have our gallbladder. The gallbladder stores bile, which will be released, and bile will help in the mechanical digestion of lipids. The pancreatic duct as well as the bile duct will merge before entering the duodenum. So this is a very important slide that shows you all the function in the aspect of digestion that is carried out in the small intestine. So there are a few secretions. Firstly is the pancreatic juice from the pancreas. It is alkaline. It contains pancreatic amylase, a protease known as trypsin, as well as lipase. So it is important for protein to be denatured in 2 pH because different protein denature at different pH. So earlier on we have our acidic pH. Now we have our alkaline pH to unwind other protein that was not unwind. Bile produced by the liver but stored in the gallbladder will help to emulsify fats. To emulsify fats means to turn bigger droplets of fats into smaller droplets of fats. This will help to increase the surface area which the lipase can add on the smaller droplets of fats. Then we have our intestinal juice that secrete maltase, sucrase, as well as lactase, as well as peptidase, which is a protease, as well as lipase. So the flowchart on the right summarizes all the reactions that you have to know. There will still be starch because the food only stays in our mouth for a short period of time. The salivary amylase would not be able to convert all the starch into maltose, which is why the pancreatic amylase, which works in a different optimum pH, will help to convert the remaining starch into maltose. Then maltase will convert maltose into glucose. Lactose will be converted by the lactase to glucose and galactose. Sucrose will be converted by sucrase into glucose and fructose. And as mentioned, some of the protein might not be digested during its stays in the stomach. This is where trypsin will digest the protein into polypeptides, and the peptidase will digest all the polypeptides as well as those polypeptides from the stomach into amino acid. While in the case of fats, bile will help to emulsify them so that lipase have a bigger surface area to convert them into fatty acid as well as glycerol. And finally, this is a table showing the digestion that takes place in the mouth, in the stomach, in the small intestine by the pancreatic enzyme as well as the small intestinal enzyme. So I strongly encourage you to take out your notebook and copy this down, as this will help you tremendously. And that is all for this lesson.